Good morning, grade 9 students. In this session, we're going to continue with the genetics part 6. And this session, we're going to talk about the testicross. What is the testicross? And we're going to make a quick revision about the testicross. This testicross, it aims to determine if the parent is pure or hybrid, if it's a homozygous or a heterozygous. This is the cross that is used to determine whether it's a homozygous or heterozygous. How? We have always the dusty cross is done between homozygous and recessive, between the recessive and the homozygous. This one it could be homozygous and recessive, meaning that it should be carrying two alleles that are recessive. For example, let's take black and white, let's say. So the black is dominant. Let's give it the capital letter. This is the black. And the white is the recessive. Let's give it WW. W small is for the white. It's the recessive. I gave you these. So if I told you what are the different genotype or genotypes for the dominant and the recessive. In order to be recessive, in order to have a black, it should be only W small, W small. This is the only option. It should be W small since it's recessive, and it should be homozygote expressed in the homozygous state. However, in the case of dominant, it should be either BB or it can be BW, since it's expressed in both homozygous and heterozygous. So if we get in the descendants here, in the descendants, in the offspring, بولادهم, if we get, please, those who are unmuted, please mute yourself. So here in the hybrid, if we get in the descendants two phenotypes, meaning that we get 50% white and 50% black. Here in the recessive, how many type of gametes it gives? It's WW, so it gives only W 100%. So this will not determine the answer. The answer will be determined according to the dominant. If we have two phenotypes, so it should give two different type of gametes. So it should be B and W. So B, it will be 50%, and W, it will be 50%. However, if we have only one phenotype, so here it gives only one type of gamete, which is the B 100%. So what determines whether the parent is homozygous or heterozygous is the offspring. If we have only one phenotype, so the dominant parent will give only one type of gamete, so it's pure. While if we have two phenotypes, so the dominant person will give two different type of gametes, so it's heterozygous. So I will repeat, the determinant whether the parents are pure or hybrid, it's used by the testicles. So we use it to determine whether the dominant person is pure or hybrid. If we have only one phenotype, as a كلهم for example, black. So one phenotype, so it gives only one type of gamete, so it's BB. However, if we have two different phenotypes, so it should be two different type of gametes, and it's BW. So this is the third type of cross. So till now, we have three different type of crosses. The first, number one, we took it is the monohybrid cross. Parents, they are pure and they differ in only one trait. So they have a difference in one trait only. So we call this cross as a testic cross. The monohybrid cross, monohybrid cross. So parents are pure and they differ in only one trait, is monohybrid. Second type of cross is the self cross. Whenever we cross F1 with itself. So F1 cross with F1. What do we call it? It's called the self cross. So till now, we knew two different type of crosses. Mono, as I said, is one. So one difference, one trait. And the parents are pure. Second type of cross is called the self cross. When we cross something by itself. Example, when we cross F1 with F1. Third type of crosses, it's the test cross. Test cross, we use it to determine whether the parent, dominant person is pure or hybrid. And you use for this one, we cross it with homozygous and recessive like we did before. So these are the three different types of crosses. Cases of heredity. First, we talked about the case of dominance. So, for example, the black is dominant over the white. So we give one symbol dominant with the capital letter and the recessive with the small letter. Last time or last week, we talked about the intermediate dominance, or it's called also incomplete dominance. What does this mean? This means that we have a trait that is intermediate, like we cross the white flower with the red flower. All the descendants, they are pink. So the color pink is intermediate between the white and the red. So intermediate, so this is called intermediate dominance. And here both alleles they are dominant and both of them they are expressed in capital letter. And I gave you the way how to answer this. Also, the third case that we're going to cover today is called the codominance. Codominance, both of them they are dominant. Co-dominant, so both are dominant. But here, codominance, meaning that both alleles are expressed. Let's give an example. If we cross a white dog with a black dog, all the descendants, all the offspring, they are black dotted with white spots, meaning that they are white and black at the same time. So we have both alleles they are found. So this is called codominance. It's very similar to the intermediate, and we said both of them they will be dominant and both of them will be capital letter, but instead of saying intermediate dominance, it's codominance. And I told my students that codominance it's cooperation between two alleles. So two alleles they are cooperating with each others. Let's have an example about this. Codominance case. Here we have a bluebird was crossed with white bird. The descendants they become blue and white together. Both phenotypes they are found. So here, if I want to give symbol, so if I told you what can I deduce from this cross? After crossing a blue bird with a white bird, all of the descendants they are blue and white. So this is the case of codominance, since both alleles are expressed. So if you want to give the symbol for the W, the white, for example, it's W capital. So here it's W capital, W capital. And if we want to give the symbol for the blue, it's B capital, B capital. Why? Because both of them they are dominant. So this is BB. It gives only one type of allele. So it gives only B, 100%. So it gives B. And here it gives only W. So it will give here BW. So the genotype will be for this BW. So it's blue with white spots or blue and white together. So if I want to give the symbol, I have to say let B capital be the symbol of the allele, which is of codominance with the white allele. And let W capital be the symbol of the white allele, which is in codominance with the blue allele. So this is the symbol. It's very similar to the intermediate, but instead, intermediate, we have a color in the middle. But here we have mixing of two colors. So this is called the case of codominance. So this codominance, okay? This is the case of physical dominance. So here we covered the three different cases: the intermediate dominance, the co-dominance, and the case of dominance. So I will repeat this. Co-dominance, both alleles they will be expressed. So both allele will be found. يعني إذا crossed black with white ما هيطلع black, or gray for example, which is intermediate, هيطلع both black and white, both colors. So this is called co-dominance. So co-dominance, it's cooperation between two. Moving now to the blood groups. Blood groups, as you know, allele A and allele B, they are the dominant, while allele O is the recessive. So if individual has genotype AO, So his blood group will be what? A is the dominant over the O. The blood group will be A. Why? If it has BO, the blood group will be B. But A and B together both are dominant. So if we have allele A and allele B, so we have blood group AB. So both alleles, they are expressed. So this is the case of codominance. So both alleles, they are expressed. So this is the case of codominance. So till now, we covered three different cases. So you have to study this and you don't have to conform them. You have to study dominance whenever one allele is dominant over the other, intermediate dominance whenever we have a color which is intermediate in the middle between the two colors, and third case is the case of co-dominance, where both alleles are dominant and both of them they are co-expression. Both of them they are expressed. So co-dominance, a new different trait appeared. So since a new trait appeared, this is the case of co-dominance. So sometimes we have parents of the same trait. They give three different. So this is the case of co-dominance. So the parents, they can be hybrid. So if we have 1 over 4, 1 over 4, and 1 over 2, so this is the combination of two alleles, and both alleles are dominant, and they should be symbolized by a capital letter. Now, let's move to activity 3, the carriers of the genetic information. If you remember, whenever we start with the genetics, we said that the chromosomes, they are the carrier of the genetic information. So let's now talk about a karyotype. What is a karyotype? A karyotype is the arranged set of the chromosomes. So karyotype, this is the karyotype. They arrange the chromosomes, they get them from a cell, and they arrange them in a figure or in an image. This image is called karyotype. Karyotype, it represents the arrangement of the chromosomes. So chromosomes, they are found and they are arranged in such a figure. This is called a karyotype. And if you remember when we talk about diploid and haploid, so we're going to go back to this. And a diploid cells, diploid, so we have two copies of each chromosome. So the chromosomes, they are arranged as pairs. According to what? According to the decreasing order of size, from the bigger to the smaller. According to the position of the centromere. Centromere is the attachment site. Okay? 3. According to the banding pattern, the bands that are found and the genes that are found on this chromosome. So this criteria you have to study. The chromosomes in a karyotype are arranged according to decreasing order of size, position of the centromere, and the banding pattern. This is the karyotype. So look, from a chromosome pair number 1. Why did I say pair? Because we have two copies. So from pair 1 to pair number 22, all of these, they are called autosomes. Autosomes, because they are from pair 1 to pair number 22, and we talked about this before. However, the last pair is called the gonosome, or we can call it sex chromosome. Gonosome, or it's called the sex chromosome. Why do we call it the sex chromosome? Because it determines the sex of the individual. The sex means whether it's a male or a female. How can we determine this? We will see. So this karyotype, it belongs to a diploid or a haploid. Every couple, for example, chromosome pair number 1, we have two copies. So whenever we have two copies, this is for a diploid, two copies. While if in chromosome number 1 we have only one copy, and chromosome 2, 3, 4 till 22 we have only one copies, so this is for a haploid cell, one copy. While diploid, it's two copies. Here in this, we have from pair number 1 to pair number 22, we call them autosomes. While the gonosomes. Here we have X and Y. Here are called the gonosomes. X and Y. Whenever we have X and Y, this is a male. So the male has X and Y as gonosomes or sex chromosome. While if we have it as XX, this is a male or a female. Let's check. So chromosomes, as I said, they are the sex chromosomes. They determine the sex of the individual. They are X and Y. If the karyotype shows two X chromosomes, يعني عندنا XX, so it belongs to a female or a girl. So all the girls, they have XX, while all the boys or the males, they have X and Y. So they have X, Y. So this karyotype, if I want to talk about it, this karyotype, it belongs to what? To a male or female? It has XY. So it's for a male. However, if we have two X's, so it will be for a female. So these notes, you have to study them. What are the autosomes? Autosomes are, as I said, they are all the chromosomes in the karyotype except the sex chromosome. They are all the chromosomes unless ما عدا sex chromosomes. And humans, we have 22 pairs. What the meaning of pairs? This is a pair. If you count them from number 1 till number 22, they are 22. 22 pairs. Why? Because everyone exists in couple. So these are pairs. Couple 1, pair number 1, pair number 2, 5, 6, 8, 13, 17, and so on. So from chromosome pair number 1 to chromosome number pair number 22, we call it autosome. However, the last one is called the gonosome. So all the chromosomes, ما عدا sex chromosomes, we call them autosomes. While the sex chromosomes, they have a role whether it's a male or it's a female. Now let's move to a pedigree. What is a pedigree? What do we mean by the pedigree? Pedigree, it's a representation of the family tree that diagrams and that shows the inheritance of a specific trait or disease. It shows the relationship between the members of the family and it shows whether they have the disease or the trait or they silently carry. Well, the meaning of silently carry, if they have it but it was recessive. This is the family tree. Always and always and always. The square or the box is for the male and the circle will be the female. Here, these are the generation. So this is generation number 1, generation number 2, generation number 3, and so on. So let's have an example. Here are the grandmother and the grandfather. Let's say the grands. Okay? Grandmother or grandpa or grandma, for example. So here the grandpa and grandmother. They gave birth to your father and your uncle and so on. And then they gave birth to you. So this, whenever you put a dash between the box and the circle, this means that we have marriage. So this man married this woman and they gave birth to these children. Okay? But this one and this one, they do not descend. This one only? Right. This one only does not descend from the family. So this is called the what? This one does not descend from the family. So this is not their brother and sisters. So this is the intruder. So هيدي جوز ابنه. Okay? So we're going to talk about the pedigree in details in this slide. Sarah and Maher, both of them, they are normal and they have a family history of albinism. What is albinism? Albinism is a disease where melanin is not produced, leading to the lack of pigmentation of the hair and skin cells. Albinism, it's a disease where you have no melanin. So here is the albino man. This man is an albino. Whenever the man has an albinism, we call it an albino. This man has a deficiency in the pigmentation of the skin, has no melanin is produced. We say albino man. This is an albino, how it look like, has white hair and the white beard and the white, for example, around the eyes and so on. And this is an albino female. It looks like this. So here, Sarah and Maher, they gave birth. We said that Sarah and Maher, they are normal. They gave birth to four children: Ahmad, Jack, Marie, and Fatima. All were normal except Jack, who turned out to be an albino. So it turned out to be an albino. Construct a pedigree or family tree representing this family. First of all, these notes are very important. Always and always and always. I said the circle is for the female. Circle for the female. So here I'm going to use the white circle for the normal and the filled circle here for the affected. So trait 1 here, for example, the normal, and trait 2 for the disease. And box, the box for the man. The box that is empty for the man with trait 1, let's say a normal. Man with trait 2, which is the affected or the disease. The symbol or the fetus or the embryo will be like this. It's like a rhombus. Whenever we find a line between a male and a female, male is the box and the female is the circle, this means marriage. However, we have two lines, meaning that we have relative mating. So they are marrying, for example, cousins or something. Whenever you have this line descending, so we have children, and whenever you have these two circles or two related to each other, meaning that we have twins. So these notes are very important. So if you want to draw the pedigree, Maher and Sarah, they got married. They gave birth. So I connect them with the line. They gave birth. I made a line down. For Ahmad is a man, so I give it a box. Fatima a circle. Jack and Marie. But Jack is an albino. So what do? You don't color it. You simply, for example, you make it like this. Okay? Or if you want, for example, you can draw a box and simply you can put scratches inside this. But you have to give the legend. This is an albino man. This is a normal woman, and this is a normal man. Without a legend, you will not get the mark. Pedigree should be drawn using a pencil, and you have to give it a title. So always any figure should be drawn using a pencil inside a box like I did here. I draw the box, and you have to give it a title. So here I constructed the pedigree. Second, they told me determine the dominant and the recessive allele. Maher and Sarah, they are normal, but they gave birth to Jack, which is affected. So the allele of disease, it was masked in the parent. It was masked, meaning that it was masked by the dominant. The dominant was dominating over the allele of the disease. So the normal allele is dominant, and the recessive allele of albinism, they were recessive. Designate by symbols. So let n capital be the symbol of the normal allele, which is dominant over the recessive allele of albinism. Let A small be the symbol of albinism, which is recessive with respect to the normal dominant allele. So N for the normal and A small for albino. Determine the possible genotypes of the parents and their children. First, I have these two. Jack is an albino, and the allele of albinism, it's recessive, so it should be only in homozygous. So it should be A small, A small. So Jack should inherit A from each of the parents, A from father and A from mother, and they should have A here and A here. But Maher and Sarah, they are normal. So they should be carrying the allele and for the normal. So an A and an A. Ahmad, Fatima, and Mary, they are normal. So sure they have the allele N, which is for the normal. But They carry either allele and another N, or they can have A. So they have two possibilities, either an N or an A. So they have two possibilities, an N or an A. How can we determine this in specific? If they give us additional information. So here he said, since Jack, port 4, is albino and the albinism is expressed only in recessive, so the Jack should be pure and it should be homozygous, meaning that's pure, and it should carry the allele of the disease in the recessive and the homozygous, which is A small, A small. Maher and Sarah, they are normal. Here you have to study the way how to answer the karyotype, the pedigree. So they should be carrying the albinism allele, but it should be masked by the dominant. So they are hybrid or heterozygous of genotype and A. Normal is dominant. So Marie, Fatima, and Ahmad could receive two alleles N from each parent, or one allele N or one allele A. So they can be N, or an A. While the last part, part 5, we ask you to perform the essential factorial analysis to verify the results of Maher and Sarah. Maher and Sarah both are normal. So if I want to start, I have to start with the phenotype. Phenotype, male is Maher and the female is Sarah. So Maher, male, is normal, and the female is Sarah, which is also normal. So both of them, they are normal. And moving to the genotype. Genotype, male should be heterozygous. You should give a Jack, which is albino. So Na crossed with Na. So here, moving to the gametes, I want to work it in proportion and not in percentage. Gametes of simple gamma. So male, it will give two different types of gametes, N and A. So 50-50, but I want to work it in proportion. So 1 over 2 and 1 over 2 for A small, 1 over 2. And here, N A also, N, it's 1 over 2, and A, which is 1 over 2. Then we move to the table of cross and table of results. In the table of cross, what do we have? We have to divide it into 2, and every box should be divided into 2 boxes. Should should be drawn using a pencil. Those who are submitting the notes or writing the quiz without using a ruler, this will not be accepted as well for this week. And also the notes should be drawn using a pencil and a ruler. Use the ruler. So N, it's 1 over 2, and A, it's 1 over 2. Here N, it's 1 over 2, and A, it's 1 over 2. So N times N, it's N. 1 over 2 times 1 over 2, it's 1 over 4. N times A, it's NA, it's 1 over 4. We have also NA, it's 1 over 4, and AA, it's 1 over 4. Moving to the table of results. So we have to fill a table of results with phenotype and genotype. So genotype, so I'm going to write the genotype and phenotype. So in the genotype, what do we have? We have NN, 1 over 4. NN, it's 1 over 4. NA, we have 1 over 4 plus 1 over 4, it will be 1 over 2. So NA will be 1 over 2. And last we have AA, it's 1 over 4. So NN 1 over 4, NA 1 over 4, so 3 over 4. If I add 1 over 4 plus 1 over 2, it will be 3 over 4. They are normal. And 1 over 4 affected or albino. So this will verify the results. Why? Because one out of four children is affected with Shujat, and Marie, Fatima, and Ahmad, they are normal. So 3/4, they are normal. So this is the case of the pedigree. So this is here, we covered the pedigree. You know how to determine if the allele is dominant or recessive according to the pedigree. And also we covered the case of codominance, which is the last case in the inheritance for this session.